So in this very class, we will start with the definition of isotype. Next is the allotype, and the third one is the idiotype. So what is the difference between isotype, allotype and idiotype? So consider this particular example. In this example, you can see we are comparing between mouse IgG1 and mouse IgM. In both cases, this is mouth, but they are two different antibody molecule. One is the IgG1, another is known as the IgM, immunoglobulin M. So if we compare between these two antibody molecules, these two antibody molecule is produced by the same mouse, but their constant region, the initial portion is the variable region, the first 100 and 110 amino acid from the N-terminus region is known as the variable region, and the remaining portion is known as the constant region. So the colored portion, this pink colored portion or this sky blue colored portion is basically the constant region. Similar thing may happen for here also. This is the constant region for the light chain, and this particular portion is the variable region, constant region for the heavy chain. So if you compare between these two constant region of two different immunoglobulin molecule that has been isolated from mouse, that is from same species, you can see although they are different antibody molecule, one is IgG1, another is the IgM, but their constant regions are basically same. The constant regions are absolutely same, so that you can say they are isotypic to each other. Iso means same. So they are similar to each other. They belongs to same kind of genus or species. For example, if you take mouse IgG1 and compare with that of the human IgGM, mouse IgG1 and human IgGM. So if you compare between mouse IgG1 and human IgGM, in that case you can definitely see a variation in the constant region. That means the constant region of both light and heavy chains of mouse IgG1 should vary with respect to the constant region of the both light and heavy chains of the human IgGM. Say it is human. So say it is human IgM. So the constant region of mouse IgG1 and human IgGM may vary significantly because they are two different genera. One is mouse, another one is human. So you cannot take mouse IgG1 into human body or human IgGM to mouse body, because in that case mouse, if we inject human IgGM to mouse body, mouse will recognize this human IgM as a foreign particle, because mouse will recognize whether they have a similar type of constant region or not. They will see the constant region is totally dissimilar, and that is why whenever you inject human IgGM to mouse cell, whenever you are injecting human IgGM to a mouse cell, mouse will recognize that IgM as a foreign particle. And similar phenomena may happen also if you inject mouse IgG1 to human cell. If you inject mouse IgG1 to a human cell, then the human antibody molecule will recognize the mouse IgG1 as a foreign particle, because the constant region of mouse IgG1 and human antibody, any kind of antibody present in human, that is IgG1 or maybe IgG, IgM, IgD, whatever it is, the constant region is different. And that is why if we compare between mouse IgG and that of the human IgG or human IgM, mouse IgG and human IgG or human IgM, so they are anti-isotypic to each other. What is isotype? Isotype determinants are constant region determinants. So we are determining the constant region only. The constant region of each heavy chain class and subclass and each light chain type and subtype are same. Okay, so the constant regions are basically same of heavy chain classes or the light chain classes. If you compare between the heavy chain or the light chain of mouse IgG with that of the mouse IgM, because both of these cases they are mouse, as a result the constant region of all antibody molecules, antibody are basically present in 5 categories: IgG, IgM, IgD, IgE and IgA, and all of these antibody molecule contain similar type of constant region. So you can easily take, say I am human, I can take blood of any other human belongs to the same category. For example, I am B positive blood group human, so I can take any blood that is also B positive. In that case, although it is a different individual, although it is a different person, but their any kind of antibody molecule is quite similar to that of mine, because mine constant region of any antibody molecule, say comparing human IgG1, so they say my IgG1 and say I am taking another person's IgG1. So these two IgG1 or IgG2 or IgG3 or IgM or IgD or Ig, whatever I am taking from another individual, the constant region of both the light chains and the heavy chains are absolutely similar, and as a result I can take that antibody molecule as self, not by non-self. So this is actually what you known as, what do you mean by isotypic determinants. So if I take any other individual's antibody, not human, it belongs to cow, buffalo, monkey, maybe rat, something like that, and if we compare between IgG and IgG, mouse IgG and human IgG, mouse IgM and human IgM, if we compare between similar type of antibody, still you can find there is a difference in the constant region, and as a result what you can say, they are anti-isotypic antibody. So this is the first point, isotypic determinants. The next topic is the allotypic determinants. So isotypic determinants means if we compare between two antibody molecule of the same origin like mouse, to define antibody molecule, one is IgG1, another is IgM, you can compare the constant region and you can find constant regions are so similar to each other, and that is why they are isotypic to each other, they are isotypic. But if it is human and if it is mouse, and in that case they are anti-isotypic to each other because the constant region may vary significantly. But the allotypic difference is that, say I can produce amylase enzyme by my saliva. You can also produce amylase enzyme by your saliva. So if I sequence that what is the actual amino acid sequence present in my amylase and what is the actual amino acid sequence present in your amylase, because amylase is an enzyme is a protein is composed of amino acid. So if I sequence what type of amino acids are present in my amylase and your amylase, or my amylase and my brother's amylase, say, in that case you can find a significant difference between one or two regions. At least one or two different amino acid must be present. This is not mutation, this is called variation. This is called variation because still it is a human. No single person looks alike. You may be differently, your voice is different, my voice is different. No two persons are alike. Maybe quite similar, except identical twins. All the persons are quite dissimilar to each other. So although we are comparing between mouse IgG1 and IgM, and we can find the constant regions are so similar, and that is why these are called isotypic determinants. But if I compare two different mouse of strain A and strain B, now we are taking two different strain of mouse. It is the mouse, okay, it is the mouse, but the strains are different. For example, if we compare between E. coli strain B and E. coli K, so two types of E. coli. Another E. coli is E. coli DH5 alpha. So simultaneously, a similar type of E. coli can have variation in their strains. So if we compare between two mouse of different strain, one is the strain number A, another is the strain number B, and if I compare the two mouse IgG1, now it is that we are comparing same antibody, mouse IgG1 and IgG1, you can find although they are IgG1, but still there are some amino acid sequence variation in the constant region. Amino acid sequence variation, as I described about, although I am producing amylase, you are producing amylase, but if we compare the sequence of amino acid present, you can find one or two position may be different. And this is not the actually mutation, it is the actually variation, more appropriately. Simultaneously, in the previous one where we are describing about what is isotypic determinants, we can say the constant region of IgG1 and IgM are so similar, and that is their constant region. But now we are dealing with the fact that although their constant regions are same, in both cases they are sky blue. Pink matches with pink, sky blue matches with sky blue. But in one or two region, amino acid sequence may be dissimilar. In that case, the third sequence, this particular area actually, this particular area, you can see a ball-like structure, a triangle-like structure, and the third one, this third structure is quite different. So one or two region might be different, and similar phenomena may happen for in this light chain constant region. You can see this particular structure and this structure is quite different. So one or two amino acid sequence may be different, and as a result, we can say they are allotypic determinants. So that is the reason, although all members of a species inherit the same set of isotypic genes, multiple allele exist for some of the genes. So this is the point, multiple alleles, they are isotypic, because in both cases these are mouse, but multiple allele exist for some of the genes. This allele encodes subtle amino acid difference. So some amino acid differences may be present and known as the allotypic determinants. Okay. For example, in human, allotype have been characterized for all four IgG subclasses. So if I compare between mouse IgG, human IgG1 with that of human IgG2, for example, human IgG3 with human IgG4, we can see some allotypic differences. One for IgA subclass is present, and one for the kappa light chains, and the one for the kappa light chains. So this is the allotypic determinant. So although they are similar type of antibody molecule taken from mouse, so they have some constant region similarity, but multiple allele maybe exist for some of the genes, and this is due to the subtle amino acid differences known as the allotypic determinants. So in the first case, we are comparing between two different antibody molecule, IgG1 and IgM of the same origin, and that is why they are isotypic. If we take mouse IgG1 and comparing between the human IgM, in that case, or say human IgG1, mouse IgG1 and human IgG1, they are anti-isotypic determinants to each other. But if I compare two similar type of antibody molecule of the similar origin, say mouse IgG1 with respect to mouse IgG1, but the only difference is that it is taken from the strain A and it is taken from the strain B, and you can see the constant regions are so similar, but at one or two position there may be some variation in the amino acid sequence, and this is known as the allotypic determinants. Now the third one, what is idiotype? So idiotype means forget about the constant region, first of all. In isotype and allotype determinants, we are considering only the constant region. But in case of idiotypic determinants, we have to consider the variable region only. That means the topmost position. This is the black colored portion, topmost portion. You have to see the topmost portion, variable region of the light chain, variable region of the heavy chain. Because we all know this particular two arm of the antibody molecule are known as the FAB portion, FAB, that is fragment of antigen binding site, FAB site. So that means there must be some sort of receptor present. Because the FAB sites are the antigen binding sites, and antigen can bind to the FAB portion, not the FC portion. And as a result, an antigen can bind only to that site when it contains some receptor. For example, you can see the receptors. Different types of receptors are present. So, a mouse can produce IgG1, and the same mouse can produce another IgG1, but you can see against antigen A and against antigen B. That means, say in day one, say today, I am injecting antigen A to a mouse. I am injecting antigen A to a mouse. So after say 4 or 5 days, their plasma cell will produce antibody molecules, say mouse IgG1. And I can find several types of receptor molecules present that can bind to that of the antigen A, because they are manufactured in such a way that antigen A can bind. They are produced against antigen A. So whenever we have isolated this IgG1 molecule for antigen A, after say 7 days or 8 days, again the similar mouse has been injected with another antigen called antigen B. Say after 8 days, I am injecting the same mouse with antigen B. Again after 5 days or 6 days or maybe 4 to 5 days, same mouse will produce another IgG1 molecule, and in this case that IgG1 molecule contains a different type of receptor, and the receptor is quite specific for the antigen B. So your B cell can produce plasma cell, and the plasma cell, B cell can be differentiated into plasma cell and memory cell, and plasma cells are destined to produce a particular type of antibody molecule, and that antibody is specific for the antigen of interest. That antigen means in the first case it was antigen A, in the second case it was antigen B. So whenever we are injecting antigen A to a mouse or to a particular person, that person will produce antibody molecule after 5 days, and the antibody molecule must contain some receptor sites on it, on the FAB portion, so that the antigen A can bind. These sites are actually called the idiotypes. Individual receptors present on the antibody molecule, basically on the FAB fragment, is known as the idiotypes, and the sum of the idiotypes, sum of the idiotypes, SUM, sum. You can see the sum of the idiotypes. The sum of the individual idiotypes are known as the idiotype of the antibody. The sum of the individual idiotypes is known as the idiotype of the antibody molecule. So the idiotype may be different for the similar kind of antibody molecule, because in this case it is mouse IgG1 and in this case it is also mouse IgG1, but this idiotypes has been produced against antigen A and this idiotypes has been produced by the antigen B. So idiotypic determinants are basically the variable regional determinants, and that will be produced during an antigen exposure, and it is basically nothing but the receptors present on the or expressed on the surface of the FAB fragment, mainly the variable region of the FAB fragment where an antigen can bind. So this is all about idiotypes.